So we are here today with Christian Cooper, a PhD student in the University of Leicester. Christian is doing his PhD in cancer studies and more specifically in radiobiology. Welcome, Christian, and thank you for being here.
Hi, Athan. Thanks for having me. It's a pleasure.
So would you mind telling us a little bit more about your PhD project?
Sure. So my PhD is in cancer studies at Leicester, as you said, with Professor Don Jones. And the project sort of focuses on the potential chemical mechanisms that underlie the clinical benefits of flash radiotherapy.
So, Christian, we have invited you here today to talk to us more about your recent publication with the title "Flash Irradiation Induces Lower Levels of DNA Damage ex vivo, an effect modulated by oxygen tension, dose and dose rate." Could you please tell us more about this recent work of yours?
Sure, Athan. Thanks. So flash radiotherapy is ultra-high dose rate radiotherapy. That's radiation delivered in a fraction of a second, and this has been seen to produce a differential between that of conventional radiotherapy at low dose rates in terms of it spares normal tissue while maintaining tumor toxicity, and so it's sort of like the golden bullet for radiotherapy, if you like.
And what is the mechanism for this observation?
So one mechanism that's been put forward is that of transient local induced hypoxia following flash, and the consequences of this are that there may be better chemical repair allowed under this transient hypoxia, and therefore you might expect lower levels of radiation-induced damage following flash. However, this is yet to be proven.
Okay. So you performed these flash experiments here in Oxford?
Yes, we met with a collaborator, Dr. Christopher Peterson, and he's helped us produce this work and performed all the irradiations for us kindly.
Okay, lovely. So you mentioned that you used an ex vivo protocol in your experiments. That sounds really interesting. Would you mind telling us a little bit more about that?
No problem. So to save time and hassle, really, instead of bringing cells from Leicester all the time, we just took a pinprick of blood and we embedded this on microscope slides. We incubated these in a hypoxia chamber at a range of different oxygen concentrations from 21% all the way down to 0.25%. And we irradiated these lymphocytes at flash or conventional dose rates. And then we also used the COMET assay to test a range of total doses and dose rates subsequent to this.
Okay, I see. So in your experiments with flash irradiation, you observed lower DNA damage assessed by the alkaline COMET assay, as you mentioned. But was this true for all the oxygen tension that you tested?
Yeah, so we saw flash-induced lower levels of DNA damage across the board at all the oxygen concentrations tested, but this only became significant at lower than 1%, more specifically 0.5 and 0.25% oxygen tension.
Okay, so how was this differential between flash and conventional dose rates at the lower oxygen tensions that you tested modulated by total dose?
So we took this differential we saw at 0.5% oxygen, and we thought, how might this be modulated by altering the total dose? And so we did the same study again, but testing total doses of 0 to 40 gray total dose. And we found that flash induced lower levels of DNA damage significantly at and above 20 gray. Okay.
So you found that flash induced lower levels of DNA damage in comparison to conventional dose rates at 0.5% oxygen concentration, and at doses above 20 gray, if I'm not wrong. So what did you do to assess at what dose rates this DNA damage reduction happened?
So we essentially performed the same study again. We used the same model, and instead of altering the dose, we just altered the dose rate this time, and we tested from 0.1 grays per second, which was the conventional dose rate we used at incremental values, all the way up to 2,000 grays per second, which was flash. And in comparison to the conventional value, we saw lower levels of DNA damage were induced following 10 gray per second irradiation, and this became significant at and above 30 grays per second.
Okay, I see. Okay, so could you conclude from your findings that the oxygen depletion may actually rationalize the DNA damage reduction in flash irradiated lymphocytes at the low oxygen tensions that you tested?
Yeah, most definitely. We saw the effect was significantly lower at low oxygen tension, but the study does fall short of rationalizing any normal tissue sparing seen in vivo. Another group has put forward that the sparing of hypoxic stem cell niches may account for this normal tissue sparing, and therefore an oxygen-related mechanism may be responsible for the differential seen between flash and conventional irradiation.
Okay, great. That is all great information. Thank you very much for sharing this information with us, Christian.
Thanks very much, Athan, for having me. I'd also like to take this opportunity to thank my supervisor, Professor Don Jones, at the University of Leicester, and my supervisor here, Dr. Christopher Peterson, at the Oxford Institute for Radiation Oncology. And thanks to our sponsors as well, at CR UK, the MRC, and the Leicester Cancer Research Centre.